Yeah, good afternoon. Welcome you all for this next lecture on the inorganic chemistry of life, principles and perspectives. In the previous class, I have talked to you about the structures of the proteins, all 4 different structures of the proteins. Then I talked to you about the nucleic acid, nucleotide, nucleoside and nucleic acid and nucleic acid structures like A, B, Z, and also talk to you about the base pairing, the Watson-Crick kind of base pairing, which indeed stabilizes the double helical structure, and then also have focused on how a protein is being synthesized by the biological system and which through a phenomena called the central dogma of molecular biology, that is DNA to mRNA to protein. And then I also talked to you about the post translational modification after the protein is being synthesized, the protein structure, tertiary structures, quaternary structures taking place. Then there are certain unwanted units which are present in that are removed, and there are some post translational modifications which are even covalent in nature, something like acetylation, phosphorylation or methylation, hydroxylation, many of these kinds of things. Also talk to you about how this C-terminal and N-terminal connectivity is being made. All these aspects are being talked in the previous class, but we have not looked at how a metalloprotein is synthesized. So, let us look at how the metalloprotein is synthesized. See, for making the metalloprotein, it is not that metal ion is coming along with the protein synthesis, as the protein is first synthesized and then metal ion is being introduced into the protein. Okay. So, there are various ways of these ones. The proteins which contain a variety of metal ions as cofactors. We have seen the iron, we have seen copper, we have seen nickel, we have seen manganese, so many different kinds of cofactors. You can call it as metal based cofactors. All these play important role in either the structure of the protein stabilizing or the catalysis of the protein. So, they are involved as a structural and catalytic ingredients of the protein as a metal center, and I have already explained to you earlier how this metal center is connected to the side chains of the protein to form a coordination complex. So, that means when the metalloprotein is formed, straight away the coordination complex is not formed. Protein is initially synthesized, then followed by introducing the metal ion into this. Okay. So, there are several metalloproteins, metalloenzymes in our body. In fact, almost 1/3, a bit more than even 1/3 of the proteins that are there in the human body are all metalloproteins. So, that means there is a huge amount of reactivity, functionality, responsibility taken by the metalloproteins and metalloenzymes. Generally, there is a feel for us to ignore the inorganic parts in the biological systems, but this particular course will bring to you how important is the inorganic part in this. There are a variety of examples: hemoglobin, plastocyanin, carbonic anhydrase. These are just for a few, and many examples we have already seen earlier, and many more will come later on. Now, let us look at how such a metal ion is entering into the protein to make the protein into a metalloprotein, or to make the protein into a metalloenzyme. So, metalloprotein and metalloenzyme. Okay. So, these are something where you can say you have your ribosomal mRNA, and which gives the peptide synthesis, which we have seen just a while ago in the previous lecture. Okay. So, and this gives the unfolded and the metal free. One of the possibilities is that the metal can be inserted. So, there are 3 ways paths shown: path going via top, path going straight, path going through this. There are 3 different ways by which we can think of the introducing the metal ion into this particular proteins. So, the protein synthesis, metal ion insertion, and then goes into the unfolded but metal bound, then goes into the total folded structure. So, the structure which are shown like this are called unfolded, the structures which are shown like this are called the folded. So, this protein is same as this protein, but structure is not the same. So, in other words, the primary structure of this and the primary structure of this is same, but the tertiary structure of this and tertiary structure, secondary structure of this and secondary structure of this, they are not the same. So, that is one way you can make the metalloprotein. So, the folded holo, holo means metal ion present, holo means metal ion present. So, folded holo protein or metal bound protein. Let us look at the other one, the protein, and while protein is synthesized in the vicinity, the metal ion is brought and the metal ion can get into the folding process, may induce the folding process and the folding will take place. So, that can also be released. Okay, and then the folding and release folded apoprotein, and then you make the put the metal ion into this, or you can take the holoprotein, metal ion removal, that is shown by this dotted line. So, if you show by this dotted line, you will get the apoprotein, and from this side the protein folding will still give the protein, but not in the metallated form. So, this is called apoform. Apoform plus metal insertion gives the final form. So, this is the final form of metalloprotein, and this is where the the protein is synthesized. So, between protein is synthesized and the metalloprotein is formed, you have these three paths which are shown. Okay. So, now what we need to remember now is that the metal ion is added after the protein is synthesized or during the protein is synthesized, which will induce the metal folding, or complete folding would have also occurred, then followed by introducing the metal ion. So, there are three different possible ways. There are three different possible ways by which the metalloprotein can be synthesized from the simple protein. Then other things that one needs to remember at this stage is the protein having the metal ion in it is referred as a holoprotein. The holoprotein minus the metal ion is referred as the apoprotein. So, hereafter in future we will be referring to proteins in the form of holoprotein, protein in the form of apoprotein. Apoprotein means the metal ion is removed. In fact, one can do that even by using some chelating agents, you can remove that too. So, why is it removed? Removed because to do certain kind of reactions. Suppose you want to reintroduce a new metal ion or introduce another type of metal ion into this. So, various things can be done in order to find whether that particular metal ion is really important or not important. So, this gives a overall feel as how and what are the different ways by which the metal ion can be introduced to make the metalloprotein. Of course, if it shows a function, it is called metalloenzyme in that. Let us look at one example here. In this particular example, it is a bit different kind of an example. The example is shown for the heme. So, for the hemoglobin synthesis. So, in the hemoglobin synthesis, there are many previous steps which we are not showing that. You take the protoporphorinolene 3, this is one other stage, and this is converted into protoporphyrin 9, and this is the form in which it is present in the hemoglobin. So, the protoporphyrin 9 is the one which is present in the form of the porphyrin that we see, and this is coming from the precursor of this. Okay. And this is still not having any metal ion in this. So, now this into this you introduce the metal ion. So, how do you introduce the metal ion? In this particular case, there is something called ferrochelatase. So, the ferrochelatase is because iron and then bringing to make the chelate formation of this, and that will be introduced into the protoporphyrin 9, and once the iron is introduced, the protoporphyrin 9 plus iron is called heme. So, we have the metalloprotein is equal to protein plus metal ion. Okay. So, the metal ion can be added at polypeptide level, or it can be added in a apoprotein. That means it is fully folded, or it can be unfolded but complete protein. So, there are different types, the different stages where it can be introduced at simple polypeptide level, or it can be at the apoprotein, that means already fully folded and regular protein is formed into which it can go, or other possibility is you have a complete protein but not folded enough to have the structure, into this as well it is possible. Okay, you have a protein plus metal ion as integral part. Of the protein. Okay. So, these are some of the things that we have looked at. In case of heme proteins, so in case of heme proteins, so heme is porphyrin plus iron. Okay. So, there are different kinds of porphyrins, and the different kind of porphyrins are present in different kind of heme proteins. So, with that we will see whenever the future discussions come on that particular thing. Okay. So, having seen this, the protoporphobilin 3 to protoporphyrin 9 to heme by using the ferrochelatase. Okay. And as you can see, finally you will form the entire protein, which is a functional part of the protein. So, where the protein in the structured form and the heme and the iron present in this. So, you can see in different ways of the same protein. In this context, that we need to remember are two aspects. One is holoprotein, other is the apoprotein. Holoprotein is protein plus the metal ion. Apoprotein is metalloprotein minus metal ion. Also mentioned to you, it is possible by using some kind of a chelators, you can selectively remove the metal ion and then to make the apoprotein. The other way, you can take the apoprotein, add the metal ion, you can get the metalloprotein too. Okay, now let us look at the other aspect of it. You have a protein, you have a metal ion, so binding through their side chains, etc. So, therefore, this is a primary coordination sphere. Okay, so that means some amino acid 5 then amino acid 20, then amino acid 40, then amino acid maybe 70, maybe amino acid 110, whatever. So, different levels of the amino acids are bound to this and forms a coordination complex. And now, as I said earlier, the properties of this metal ion N plus is very much different from the metal ion in water or the metal ion in any of the in vitro kind of a systems, because the protein applies its own conformational restrictions, its own the hydrophobic hydrophilic regions influence, and thereby it influences the total properties including redox properties of the metal centers of these ones. Okay, now, if you want to claim this AA5 is really bound, how do we know that the AA5 is the one of the directly bound amino acid in this? AA5 must be there in the primary structure, there is no doubt, but how do you say that the AA5 is bound or AA40 is bound to metal center? The simplest way is you change this AA5 from one whatever be the amino acid there to a different kind, and then look at whether this protein is stable, whether the protein is functional. So, if the answers is yes, the protein is stable, protein is functional, functioning the same way as that, that means certainly AA5 is not bound. On the other hand, if your answer were to be that the AA5 when you remove this into a different amino acid, because the different amino acid do not have the same kind of a side chain, therefore, will not be binding the same, and the rate reaction catalysis will not be the same because you have changed the binding partner, which is not binding also in fact. So, in such a case the function will change. If both the stability and the function of the protein changes, so obviously you can say that particular amino acid is absolutely essential. So, how do we do that kind of a change? So, to do that kind of a change, changing one amino acid to other is referred as the mutation. It is called mutation. Okay. In fact, in the previous example, I can change this by changing the corresponding codon. I can change this by corresponding codon. Codon is a triplet pair, triplet from the corresponding nucleotides. I can change this one, I can change this one, or I can change more than one. So, therefore, if I change one at a time, it is called single mutation. If I change more than one, it is double or multiple mutations. So, mutations could be single, double, or multiple. So, I can change only codons for one amino acid, I can change codons for two of them, I can change the codons for several of them. So, this mutation. So, by doing this I will get a protein with a change in the amino acid in that particular position. So, let us look at the next slide in this connection. So, you can do so making specific changes in DNA sequence by the mutation. Okay. So, you would like to change that should reflect in the protein synthesis. Okay. This is this goes through different processes called sequence insertion, sequence deletion, sequence addition. I am not going to go into the details of all this, because these kind of things you will study if you have interest in molecular biology course. But all that I would say is all these are the kinds of operations that need to be done with the DNA. So, those operations, so just see, suppose you are looking at the single mutation, one mutation, and it is at a particular position, it is called site targeted mutagenesis. okay, at a particular site. You target at a particular site. So, this is your genome or DNA part of it, and you are interested in this particular codon or region. So, that is what is referred as the gene X, whatever we think is that, and you select this one, and this is ready now. Then you take a plasmid and you cut the plasmid here, with this line and this line, and you got a open plasmid. So, from a circular plasmid you got a non-circular plasmid or a linear plasmid, and this piece is out. In the position of the piece you put this guy. So, this gene what you got. So, this gene should have information what you wanted to synthesize, that triplet code should be there. So, you want to synthesize a particular amino acid, you want to replace a particular amino acid in the previous case, then you cut that port and put this new piece. So, now it is integrated into this. Now, so you have the plasmid, you have integrated the whole gene with the modification, and that is now is put into the some live bugs, for example E. coli. So, you can introduce this into E. coli and express the system, then you will get to express the protein. The protein that comes out will have the modification in this particular thing. So, in a site targeted mutagenesis case, you will have, so one with this process you will get at that particular site the amino acid is changed at that particular site. And this is referred as site targeted, it's called site targeted mutagenesis. So, once you synthesize this can be synthesized by using the E. coli cells. You put this plasmid into the E. coli. So, these whole operations are called genetic engineering operations. What I have mentioned is very little portion of this, at the tip of the iceberg, not anything details. Somebody who is interested in these more details, they should approach a biochemistry, molecular biology course more precisely speaking. Okay. So, you have a genome, you select the genome where you have this particular modification is there, and then put this the gene integrate into a plasmid which is already cut, and this plasmid which is integrated with this gene X is introduced into the live cells, and then allow the expression to take place, okay, and that will give the protein synthesis aspects as well. Okay. So, thus in this entire process of what I have been doing in the last several hours of lectures is that elements which are of essential elements, okay, and of course, we have looked at the essentiality. The essentiality is looked at, and we have worked out all the criteria. So, essentiality and deficiency and excess syndrome. Then we have looked at the element or ion absorption by intestines. We looked at mutual interactions between the elements absorbed Okay, this is antagonistic and synergistic. Okay, then we have looked at the necessary dosages, okay, of essential trace and ultra trace. Ultra trace elements. Then we started looking at how these are bound. And prior to that, we have also looked at why these elements and not the other, why these elements are so specific, why only these elements are chosen by nature. And there we have looked at variety of coordination characteristics, stabilities and lability aspects are important. And I am still to explain this stability lability, which I will be doing in the next lecture in any case. So, and coordination characteristics, stability, and then we talked about how these are bound through the side chains, how these are bound through side chains, and then we brought the concept of the metalloprotein as a metal ion complex inside a protein, metal ion complex inside a protein. And then we try to say that the simple metal ion in water versus metal ion in protein. These are absolutely different. The properties of the metal ions are very well modified by the protein as compared to the metal ion, and that is why different proteins would bring different kinds of reactivities to the metal ions. Therefore, we have a huge variety of metalloproteins and metalloenzymes functioning in this. Then I have taken to explain you these amino acids, proteins and peptides, amino acids, proteins, okay, in between peptides and proteins and their structures. And we have also looked at nucleic bases, nucleosides, nucleotides and nucleic acids and their structures. So, we have looked at all of these. Then we looked at the protein synthesis, and then we looked at the metalloprotein synthesis, where the metal ion is introduced into the protein that is being synthesized and then brings the property in different various ways. And also now we talked about the Holo protein versus apoprotein protein. Then we have talked about the mutagenesis. So, okay, these are all various aspects that encompass us to understand the basic conceptual ways of this role of inorganic elements in biological systems in the form of inorganic chemistry of life. And in the next class I will talk to you about the stability, lability, reactivity, all of these aspects are the metal ions when they are there. So, far we have only looked at the biological side, then we will look at the inorganic chemistry aspects of side. Thank you very much.